So for today, I will discuss about mutation and gene regulation. So what is mutation and what is gene regulation? So let's find first what is mutation. A mutation is an alteration in the nucleotide sequence of the genome of an organism, virus, or extrachromosomal DNA. Viral genomes contain either DNA or RNA. A mutation is a change in a DNA sequence. Mutations can result from DNA copying mistakes made during cell division, exposure to ionizing radiation, exposure to chemicals called mutagens, or infection by viruses. Germline mutations occur in the eggs and sperm and can be passed on to offspring, while somatic mutations occur in body cells and are not passed on. So next is, what is gene regulation? A gene regulation is the process of turning genes on and off. During early development, cells begin to take on specific functions. A gene regulation makes cells different because these different patterns of gene expression cause your various cell types to have different sets of proteins, making each cell type uniquely specialized to do its job. For example, one of the jobs of the liver is to remove toxic substances like alcohol from the bloodstream. So, how does gene regulation relate to mutations? They include mutations in transcription factors and signaling proteins, mutations in factors that regulate the methylation of DNA and associated histone proteins, mutations in the protein complex cohesin, which regulates chromatin structures, and mutations in proteins involved in splicing. Mutations are abnormal changes in the DNA of a gene. The building blocks of DNA are called bases. The sequence of the bases determines the gene and its functions. Mutations involve changes in the arrangement of the bases that make up a gene.